So another way that prokaryotes are said to be more similar to each other than they are to eukaryotes is that prokaryotes are generally said to lack internal membrane systems or membrane-bound organelles like you see in eukaryotes. And so in addition to the nuclear envelope that you find in eukaryotes that demarcates the nucleus, eukaryotes also generally have mitochondria and or chloroplasts or a whole other range of organelles. And archaea and bacteria are said to completely lack these. However, we now know that this isn't the case, and the planktomycete group of bacteria includes many examples of this. In figure A here, we have the most simple example, where you have additional compartments within the cell besides just the cytosol. So in this case, you have a peripoplasm, which is said to be the portion of the cell that lacks ribosomes. Within this peripoplasm, you have a secondary membrane, and within this membrane, you have what's known as the parallelosome. Parella is a genus of bacteria where this was first identified. Now, within this parallelosome, you have ribosomes, and you also have a nucleoid. Now, a nucleoid is not a nucleus. A nucleoid is just a condensed form of DNA often found in bacteria. And so there's no nucleus in this bacterium, but there is a membrane-bound compartment that includes all of the genetic material. Now, in figure B, you can see this goes even further, and that this secondary internal membrane actually has an invagination and a second compartment in which the nucleoid is kept separate from the rest of the cell. And so in this case, you have the peripoplasm like you have in figure A, where you have no ribosomes. You then have this parallelosome, which contains ribosomes, and you have a secondary parallelosome contained within a nuclear envelope. And so this is directly contradictory to this idea that prokaryotes lack internal membrane systems. And there's even a further example of a membrane-bound organelle within this parallelosome, in the anammoxosome, which is found in Anammox bacteria. Now, Anammox just stands for anaerobic ammonia oxidation, and that's kind of a niche metabolism that was not very well understood for a long time until this species of bacteria was identified with the anammoxosome. And so this has the same benefit as other organelles in that by concentrating all the enzymes and substrates required for this metabolic process, you can increase the efficiency and you can increase the how favorable it is energetically. So before I go on the note of ribosomes, I think it's important to break down how these Fedberg units actually work. If you actually look into it, you'll find that ribosomes have a small subunit and a large subunit. In archaea and bacteria, there's a 30S small subunit, a 50S large subunit. These come together to form a 70S functional subunit. And in eukaryotes, you have 40S and 60S coming together to form an 80S functional subunit. Now, these don't add together in any intuitive way. Obviously, 30 plus 50 is not 70. 40 plus 60 is not 80. And this is a result of the fact that sedimentation rates depend on multiple variables that are not related in a simple manner. And so the sedimentation rate is not directly, it's not a direct result of the mass, and it's not a direct result of the structure of the subunits. And so it's really not very useful, I think, to try to say, well, bacteria and archaea both have the subunits that fall out of suspension at the same rate, because it's not related to the function or even the structure of the subunits. And so don't get confused by this and start thinking that archaeal and bacterial ribosomes are more similar to each other than they are to eukaryotes. They're not.